I'm immensely honored to be asked to give this talk. For many years we've known about the work in Colorado and have admired it, but I'm especially pleased because we're able to interact with students who might carry this work on in years to come, and that's a special treat for me. In our own program, we've not done plant breeding for drought tolerance and have instead explored the principles governing the plant response to limited water. The hope is that awareness of the principles might help the plant breeding effort. Today I'll discuss some of the biochemical and molecular aspects of this work that we've done, developed from field experiments that I talked about yesterday. We use maize as our model, and we'll focus on today on how grain number is controlled. Losses in grain number are particularly serious because they are generally irreversible. Grain development ceases and the florets are said to have aborted. By contrast, losses in grain size are often reversible if water is resupplied. We discovered that we could prevent abortion by feeding the plant intravenously while photosynthesis is inhibited by drought. We started by feeding complete nutrient medium for embryo culture that had been developed by Burl Gengenbach at Minnesota. As long as we fed photosynthetic quantities to replace the photosynthesis that was not occurring, we could prevent abortion. We found from analysis that sucrose was the active ingredient in the Gengenbach medium. We called this reversion functional reversion to distinguish it from genetic reversion, which is well known to all of you. But like genetic reversion, this functional reversion can help to identify the genes that are controlling the phenotype. And that's the effort that I'd like to describe today. First, the problem. Water deficits cause floral abortion in crops that reduce the number of reproductive structures. Water deficits affect the expression of most genes. Water is, of course, involved in almost everything that the plant does. And Zinselmeyer and his colleagues at Pioneer, doing work with microarrays, showed that hundreds of genes are affected around the time of pollination when the abortion effect occurs. Tim Setter and his colleagues at Cornell did a similar study and again showed hundreds of genes were affected by water limitation in maize at this time. And consequently, our problem is identifying which of these genes actually are controlling the abortion that we're seeing in the phenotype. And that's what we'd like to explore today. This lady is about 1.5 meters tall, or about half the height of a maize crop that has been supplied with water, and this is a characteristic of drought in our area, which occurs with these kinds of effects about 1 year out of every 2. In this particular year, drought occurred at the time of pollination, and you can see the plants reached about 50% of their usual height, and on this ear there's about 15% of the usual kernel development, the rest having aborted. Normally, of course, there are about 500 to 600 kernels on an ear that's developed normally. And if we think about the life cycle of maize, we can divide it into basically two two aspects as we can for most crops. First, vegetative development, which occurs until about 40 or 45 days after planting, and then reproductive development that occurs thereafter. The abortion window for this crop is right in this region from about 40 or 45 days to about nearly 60 days after planting. Meiosis is occurring within this window, and also pollination would normally occur. We've been studying, focusing on this area because, as I mentioned, the effects tend to be irreversible, whereas water limitation that occurs after 60 days tends to result in smaller kernel size rather than abortion. And it's reversible because when rain or water is resupplied, then the grain can grow again as photosynthesis resumes. Our feeding method involved supplying sucrose, the active ingredient in the gingimbach medium, to the stems of these plants. And because we could demonstrate that water deficit around the time of pollination caused abortion within a window of about 5 days duration, we withheld water for 5 days from plants growing in soil in controlled environments where we could achieve agronomic yields at field planting densities to try to relate as closely as we could in these environments to the agricultural environment and system. Withheld water for 5 days and then re-watered. And in that time, irreversibility of abortion occurs. When we fed sucrose to the stem and fed for all 5 days to replace the sucrose that was missing from the lack of photosynthesis that occurred during the drought. We fed sucrose here in this internode by creating a well in the stem and attaching a reservoir that contained the sucrose solution. But because the plant seals this wound after about 6 to 12 hours, we had to repeat this process each day. And so the second day we fed the next internode, the third day the next internode, and like that. Because only a few days of low water potential and low photosynthesis trigger abortion, we had to look at the behavior of these plants as we withheld water from these pots during this 5-day period. And here's leaf water potential behavior and also the behavior of net photosynthesis. That is, the photosynthesis above and beyond the respiratory requirements of these plants. And what you see is the water withheld on these days represented by the black bar, sucrose fed to the stems shown here by the white bar, the controls shown by the open circles, the low water potential treatment shown by the closed circles, and then the sucrose fed behavior of these plants, where sucrose is being fed to the stems, shown by the black squares and the dashed line. And I'd like for you to remember this kind of format because I'll come back to it over and over again in this talk. Okay, and you can see that the feeding of the sucrose solution did not change the water status of the plant. This is because the amounts that we're feeding are much less than the amount of water that's actually flowing through this plant even during the drought. It also, the feeding did not alter the photosynthetic activity. Okay, the functional reversion to which I referred earlier is shown here. This is the yield of controls on the left. You can see the ears, about 500 kernels per ear. The abortion that we achieved during this 5-day treatment that I just showed you is shown by the ears on the right. You'll notice they're deformed. That's because on the back side, which you cannot see, we have sampled these ears and used the samples in the slides I'm going to show you subsequently for many of the experiments. You can see that normally we have about 500 kernels per ear. The abortion at the end of the season has resulted in almost 0. So we have a very large effect here, and the question is, what happens when we feed these plants? And that's what happened in this particular experiment. We achieved somewhere around 70% reversion. The conclusion from all of this work is that sucrose fed to the parent plant in photosynthetic quantities prevents abortion. Now, we're feeding to the stem, it's preventing abortion in the ear. So now what I'd like to do is look at what the physiology and biochemistry is that's involved in this reversion. We're going to look mostly at this biochemistry. Sucrose, as it arrives at the ear, is processed, we think, first by cell wall acid invertase. And acid invertase hydrolyzes sucrose into its component sugars, glucose and fructose. There are then several steps, and some of this glucose ends up as glucose chains in the starch molecule, which is stored in the floret that I will show you. What we're going to deal with here is first sucrose and what drought and feeding does to that sucrose in the female floret of the ear, because it's the female, from cross-pollination experiments we've been able to identify, it's the female that is blocking development of the female floret, is responding to the low water potentials. Okay, we'll look at sucrose in the female floret, specifically in the ovarian part of the female floret. The acid invertase activity, glucose, which is the product of that enzyme, or one of the products, and starch. So we'll look at those 4 aspects of the biochemistry of these structures. First of all, what about sucrose in the ovary portion of the floret? We can see here that drought has resulted in a decrease in sucrose content of the ovary, shown on a per ovary basis. So you can see the dark circles here, which are the water-limited plants. Sucrose levels per ovary is dropping through this 5-day period. And in the controls, of course, the sucrose content per ovary has gone up. So there's a big difference in the sucrose content. If we feed sucrose to the stem, it has an effect on what's going on in the ovary of the florets of the developing ear. And you can see that we're able to recover or to reverse the sucrose that is missing in the dehydrated plants by this feeding process. In fact, it looks as though we've completely resupplied the sucrose that would have been missing. And of course, these plants that we're feeding sucrose to are a fraction of the dehydrated plants. Now let's look at invertase activity. What you see here is invertase activity shown in pictorial form with the quantification of invertase activity in graphic form below. And you can see in the controls here that the ovary, from which the stigma and style have been removed. In other words, the silk portion of the female floret has been removed. So we're just looking at the ovary, and the nucellus is visible here. In this structure right here labeled N, and that's where the embryo sac is located. And you notice this is 5 days before pollination, so there is no embryo. This is totally maternal tissue. Just below the nucellus is a pedicel where the phloem termini are located, and the phloem does not enter the nucellus directly. So the phloem has to deliver its contents to the vicinity of the nucellus, and then we'll see what happens to it. Notice that in these controls shown in this upper series of pictures, the ovary is getting larger and larger, as you would expect. Here's the ovary on the day of pollination, and on that day you can see there's a dark stain here. That dark stain represents the cell wall bound acid invertase activity in this ovary. It becomes more intense as the ovary gets larger. Notice down here in this row, which would be the water deficient plants, that development of invertase activity does not occur. On the other hand, if we look at the sucrose-fed plants, you can see that there's a partial recovery or prevention of the loss of that invertase activity. Quantifying that in this graph down underneath, you can see that the controls had gained in invertase activity. The water-limited plants down here at the bottom of the graph did not, and we've been able with sucrose feeding to partially reverse the activity of the cell wall bound invertase in these structures. Now, John McLaughlin was in our lab at the time, and he developed a method to look at glucose location in these ovary structures. The glucose is high in the red and low in the blue color. And what you see here again is that row of ovary structures at the same time as we were talking before, and just below the structure is the autofluorescence of that same structure. So you can see the glucose, and you can see the vascular system that is present in that structure, in this autofluorescence. And you can see it fluoresces white, and then there's a sort of a cup structure. A cup structure just below the nucellus, almost like a golf tee with a ball sitting in on the tee. What you see is that early on, before pollination, about 5 days, no embryo is present. There's some glucose there, but it's pretty minimal. You can see that as sort of a green haze. But by the time we're 3 days later, glucose is clearly present, and it's abundant, accumulating in the around the phloem termini, and there's now glucose up in the nucellus. And there's a steep gradient from the red to the green, favoring transport of glucose from the phloem termini into the nucellus. And that intensifies as this ovary becomes larger and larger in these female florets. If we look now at the water-limited plants, you can see below here in this row that while there's a little bit of glucose here in this first one, at about -2 days, it completely disappears in that structure as this drought becomes more intense. And if we feed sucrose to the stems, then the glucose in the ovary of the ear is partially returned to the control level. You can see the quantification below in analytical terms, and you can see that it reflects what we've seen previously with respect to the invertase activity in the previous graph, and also what you see up here in these panels. That is to say, we've achieved a partial reversal of glucose content in these ovary structures. Now let's have a look at the starch. Now we're looking downstream in the biochemical system that we're seeing here, and you can see that starch is abundant in the pedicel tissue around the phloem termini, but there is no starch in the nucellus. The starch here, of course, is stained by iodine and is the dark color that you see. As the ovaries get larger, you can see that starch remains, and there's still quite a bit of starch here 2 days after pollination and fertilization of these the egg in these structures. If we look at the bottom row of figures, you can see that the starch is present on day -2 as the drought is just becoming serious, and it disappears on day 0 at just the time that the glucose disappears. Now bear in mind that the starch is long chains of glucose, and it's being broken down to supply glucose to this structure. As I pointed out yesterday, the parent plant on which these structures are developing has reserve, mostly in the form of sugars, which could keep these structures growing, but it refuses, at least in our environment, to give that reserve, photosynthetic reserve, to these growing structures. And consequently, these structures are on their own and have to depend on the reserves that are stored in the structure itself, in the ovary. And when that reserve disappears, as you can see here, then this structure is deprived of substrate for its basic metabolism. And the glucose disappearance and the starch disappearance coincide. If we feed sucrose to the stems, what you see is that part of that starch doesn't disappear. And you can see that in the graph underneath where this is all quantified. So to summarize, what we've looked at is various steps in this biochemical process, and sucrose delivery, because of the inhibition of photosynthesis, is blocked. But cell wall acid invertase, you notice, does not come back all the way. And all the succeeding steps, the glucose, which we saw, and the starch steps, we're able to reverse partially so far. But we have not so far been able to reverse it fully. And that's, I think, why we saw only reversion to about 70% of the kernel development, whereas our controls, as far as kernel number was concerned, we had a measure of the full 100%. So there are 2 blocks here, one of which we can reverse completely. That is sucrose, with our sucrose feeding. But we have not so far achieved complete reversion of cell wall invertase, but we can achieve partial. Okay, so in summary, the lack of photosynthetic reserve starves the female floret, seems to trigger abortion after the starch is depleted in these structures. Do the florets simply starve, or is there active gene regulation involved in this process? And now what I'd like to do is to turn to the gene regulation that is going on at the same time. As I mentioned, hundreds of genes are being changed at this time. Our problem is to find which ones are controlling this abortion phenotype. The phenotype is very obvious in this particular experimental system. So we're now looking here at the cell wall invertase gene family. There are 4 cell wall acid invertase genes in maize. So it's a 4-gene family. 2 of those are not expressed at this stage of development to any significant degree. But 2 are, cell wall invertase 1 and cell wall invertase 2, which are graphed here. Now these are being measured with real-time fluorescence, with real-time PCR. And that's how we did these time courses through this experimental period that I mentioned earlier, or described earlier. What you see is that cell wall invertase 1 increases in gene expression as these ovaries grow, and this is relative to the expression that we see on day -5. That is 5 days before hand pollinating these ovaries. And in the controls, you can see gene expression goes up, but there's significant downregulation in the dehydrated plants. And in this member, cell wall invertase 1 does not respond to our sucrose feeding. And so this one is not sugar responsive. It's drought responsive, but not sugar responsive. Cell wall invertase 2, on the other hand, shows the same downregulation, but when we feed sucrose, we can completely reverse the downregulation of this gene. Now, there's a very curious thing going on here, of course, in that this, the expression of this gene shows this curious dip on the day of pollination. We found this in all 3 replicates of this that we did for this graph, and we don't know why that occurred, but it's there. But on the other days, you can see that there was a specific downregulation of the expression of this gene for cell wall invertase 2, and it was fully reversed by feeding sucrose. We think that the lack of response of cell wall invertase 1, but the full response of cell wall invertase 2 to sucrose feeding could probably account for why we saw only partial reversion of cell wall invertase activity, and also all of the succeeding steps in our sugar-fed plants. And we've looked at all of the genes that are responsible for sucrose processing in these ovaries. There are 9 of them. We've looked at all of them. All of them are downregulated by water limitation, except for one, and that's the inhibitor protein for invertase, and it is unaffected by drought, unaffected by sucrose feeding. There are, among these 9 genes, there are 4 which are sucrose responsive. This cell wall invertase, there's a soluble invertase which is located in the nucellus but not in the pedicel. That's also sucrose responsive. The sucrose responsive ones, because they reverse the phenotype, must be the genes that are involved among these 9 in the control of the phenotype. So we can basically ignore the genes that are not responding to sucrose, with some exceptions. Now, as I mentioned, the sucrose, the abortion is irreversible. And so we know there is an irreversible aspect to ovary development at this stage, which somehow is lost at a later stage of development when we simply see the grain becoming smaller, but the grain is nevertheless quiescent during a drought. And when we re-water, growth resumes. So this aspect of what we're looking at is an attempt to find what accounts for that irreversible step in ovary development when abortion takes place. And what we see here are 2 examples of senescence genes among 4 that we looked at. These 2 showed upregulation in contrast to the downregulation that we saw before, indicating that there's active involvement of the genome in the abortion process and the irreversibility of it. And these 2 were upregulated. The other 2, which were DNase and ribonuclease encoding genes, did not respond to the drought and did not respond to sucrose feeding. These 2, on the other hand, ribosome inactivating protein number 2 is a member of a 2-gene family in maize. Ribosome inactivating protein 1 is involved in programmed cell death of endosperm at the maturation of the seed. Most of you will know that programmed cell death occurs in the endosperm, causes breakdown of the membranes of the endosperm, which is now loaded with starch as the seed matures. And this allows enzymes to reach the starch during germination. So programmed cell death is very much involved with ribosome inactivating protein number 1 in this gene family. Ribosome inactivating protein number 2 has an unknown function. It inactivates ribosomes by removing a purine from a specific position, and it has a global effect, and that's why we were interested in this as a possible senescence-inducing enzyme. And what you see is that on the day that the glucose disappears, this gene is massively upregulated. It's 100-fold upregulated, and not only that, but if we feed sucrose, we can prevent that upregulation, and that's shown there in this left-hand graph. You see that the dashed line is right down there with the controls, and the massive upregulation you see on day 0, the day of pollination, is occurring just on that day when the starch has disappeared and glucose has disappeared in these ovaries. Now we re-water, and on day number 2 is a day after we've re-watered, and you see that ribosome inactivating upregulation is now dissipated. And I might go back and just mention that's in contrast to what we saw here in the sugar responsive genes. You see that on the day that we rewatered, day number 2, there's evidence that they're coming back in activity. You see the black dots are now higher than they were before. So it appears that the reversibility of invertase activity upon rewatering is present. It can be observed in these genes, and the same reversibility shows in this ribosome inactivating protein. But notice that later on day 2, even though the plants have been rewatered, senescence has apparently been set in motion, because on day number 2, despite the fact the leaf water potential has recovered, photosynthesis has recovered, these plants are showing an increase in phospholipase D1. Now, phospholipase D1 codes for an enzyme that breaks down cell membranes, plasma membrane in particular, and you can see that it's upregulated on day number 2. So after phospholipase after ribosome inactivating protein number 2 has had its effect, then we see this upregulation of this other gene. And this one also is sugar responsive. That is to say, we can at least partially prevent it. Well, do we see evidence of membrane degradation? John McLaughlin used this Evans blue stain to detect losses in membrane integrity, and this is a shortened version of what I've shown you before, just on the last couple of days when this upregulation for senescence is occurring. And what you see is that the cells are staining here in the dehydrated plants that have nevertheless been re-watered. And what we see in the lowest diagram here is just a magnified view of what we see up here in diagram F. So this one, when magnified, is showed down in the bottom figure. So this is a cell-specific stain that is entering the cells and staining the cell contents because it can go through the membranes, which have apparently lost their integrity and now can no longer prevent the entrance of the stain into the cytoplasm. Whereas you can see that in the controls there is no staining. There's no staining here on day number 2 or on the day of pollination, and in the sucrose-fed plants we've been able also to see no staining. So it appears that induction of senescence has had an effect, that is, that upregulation of gene expression has had an effect in this particular system. In summary, then, the way we're thinking of ovary abortion in these florets is that at high water potential, there's high photosynthesis, normal photosynthesis, and there's starch present in the ovary structure itself. So later on, when low water potentials inhibit photosynthesis, the parent plant is unable to provide sucrose to these developing structures, even though there's sufficient sucrose in the plant to keep these structures growing. It does not transport it. Now, I should mention that later on, when there's even more sucrose in stems and leaves, this will be transported to these developing kernels. But right now, at this stage of growth, we've not been able to see that. Now, as a result, there's a downregulation of sucrose processing genes pretty much across the board. Some of these are sucrose responsive, some of them are not. But what happens because the parent plant is not providing sucrose to the developing floret is that the floret now is relying on its own starch, and that starch is supplying glucose for a while, but after a while becomes depleted. We see the disappearance of glucose, disappearance of starch, and now that causes an upregulation of genes of senescence. And as a result, senescence occurs and causes abortion. So we are thinking that these latter steps are probably the causes of irreversibility. If they were not there, in other words, this system would be quiescent, just as happens a couple of weeks later during a drought, when those developing kernels will not abort, or abortion is much less apparent. So in summary, around the time of flowering in maize anyway, there are hundreds of genes that change in expression when water becomes limited. Our problem is, how do you account for the phenotypes? Which genes are controlling the phenotypes that we're looking at? Among the hundreds of genes, functional reversion helps to identify some of those genes, and what it has done is to identify a total of 4. 2 sucrose processing genes, which are downregulated, and 2 senescence genes so far. The 4 include senescence genes that respond to sugar supply, probably glucose. The irreversibility is a later step in this whole process. It required a few days, and it wasn't until sugars were depleted in the developing structures that that the senescence genes were triggered. Well, that's what we know about this system, and I just want to say that the people who did the work are shown here in a list, and I particularly want to indicate the work, or highlight the work of Mark Boyle here at the top of the list, who developed that sucrose feeding system, and John McLaughlin, who developed the glucose imaging system, and a number of other aspects of this study. And John is shown down here with our psychrometer with which we measure water potential. So thank you very much.